Hello again, and welcome back to the Georgia Tech iGEM Synthetic Biology video series. In this installment of our lab series, we will be discussing chemical and electrical transformations. Transformations result in the genetic alteration of a cell when the cell directly uptakes and incorporates exogenous genetic material. For chemical transformation, the cell must be made chemically competent before transformation can be performed. This competence is achieved by using calcium chloride solution to alter the cell membrane permeability. The genes are added to the competent E. coli cells, and the cells are then isolated on ice for about 15 minutes. This isolation step allows the plasmid to come into close contact with the cells. The E. coli cells are then subjected to heat shock in order to disrupt the cell's membranes and create pores in the membranes for the uptake of the plasmids. The heat shock lasts for 1 minute. The cells are then placed back on ice in order to retain the plasmids in the cell. LB media is then added to the transformed cells so that the cells can grow. The cells are placed in a shaker at 37 degrees Celsius to allow the cell to pre-grow and amplify. These cells are then plated on ampicillin plates and incubated at 37 degrees overnight. After 24 hours, if the transformation was successful, there should be colonies on the plates. Transformation efficiency decreases when increasing plasmid size, so electroporation, or electric transformation, is necessary for the uptake of large plasmid DNA. Electroporation does not require chemically competent cells or any salt washes. Electroporation is easy and rapid, and a large number of cells can be transformed at one time. However, there is substantial cell death. Therefore, more cells are needed for electroporation in order to grow a sufficient amount of transformants. Electroporation still takes place on ice, but instead of a heat shock, the E. coli cells are subjected to a strong electric current. This electrical pulse creates temporary pores in the cell membrane due to the difference in electric potential across the membrane. The charged DNA molecules are driven across the membrane and into the E. coli pores. When performing electroporation, the pulse length, buffer, and voltage must all be taken into account before completing the protocol. Transformation is an invaluable technique in order to replicate target genes. The E. coli grows and replicates, thus replicating the target genes that were introduced into the cells through transformation. This allows for more DNA to work with in future experiments.